Welcome to our review of some basic chemistry that's necessary for understanding the physical nature and the chemical nature of microorganisms. This is BIO2020, Microbiology of Infectious Diseases at PLNU, and I'm Dr. Cummings. Right now we're going to talk about the nucleic acids as critical macromolecules in the cell. We'll define what nucleic acids are, the different types and the different roles that they play in the cell. Then we'll look at their monomers, the nucleotides, and we'll compare the RNA nucleotides from the DNA nucleotides. And we'll see how they come together, particularly in DNA, to form a double helix following the base pairing rules. And then we'll briefly consider the amazing diversity of DNA sequences that are possible using long genes, long polymers, that have a four-letter alphabet, so to speak, if we think of it from the perspective of a language. So these are the RNA and DNA polymers of nucleotide monomers. So when we take nucleotides, which we'll see in the next slide, and string them together by dehydration synthesis, we can form either an RNA molecule or a DNA molecule. So RNA is ribonucleic acid, DNA is deoxyribonucleic acid, and we'll see the slight difference between the nucleotides in those two. The main purpose of the nucleic acids is to store and ultimately convey information. DNA is an information storage compound, and messenger RNA in particular is critical in relaying that information so that it can be used. There are a couple other RNAs, in particular transfer RNAs, that are involved in actually translating the messenger RNA into protein, and ribosomal RNA that's part of the ribosome that is also involved in that translation process. So all the three main types of RNAs are involved in conveying that information, and the DNA is the repository. A metaphor that's sometimes helpful is to think of the DNA, the genome, the collection of genes, as sort of a big recipe book. And each gene is a single recipe for how to make a protein. That's almost entirely true. There are a few examples where they don't make a protein. The final product is an RNA molecule, like with tRNA genes or rRNA genes. But the majority of these genes are what we call structural genes that code for a protein, and they're really a recipe for how to make that protein. The messenger RNA then is just an intermediate. The DNA is the cookbook, so to speak. The messenger RNA would be like if you opened the cookbook, found the recipe you're interested in, and copied it down onto a little index card. And then you put the cookbook away in the nucleus for safekeeping, and you take the index card, the messenger RNA, back into the kitchen, and you cook up that recipe. And when you're done, you can discard that index card and be done with it. And those messenger RNA transcripts, we call them, often only last a couple of minutes. And so if the cell wants to make more of that recipe, it has to go back to the recipe book and copy down the recipe again. So nucleic acids are all about these recipes for making proteins. And we'll see in the final video of this series that the proteins are really the business end of these chemicals, of these macromolecules. They're the molecules that do something within the cell. Now we've got two different classes of these nucleotides. We've got the pyrimidines and the purines, and their sugars slightly differ also between RNA and DNA. So you can see in DNA, let me get a pointer here. In DNA, carbon number 2 has two hydrogens coming off of it, whereas in RNA, the sugar has a hydroxyl coming off as well as a hydrogen at that same position. Otherwise, they're identical. So we've got ribose, this 5-carbon pentose sugar, with a hydroxyl group coming off carbon number 2. If we remove that hydroxyl, we have deoxyribose, and that's where it gets its name, deoxyribose. So we've got ribose that's missing an oxygen. It's really missing a hydroxyl group from carbon number 2. And that slight difference is enough for the enzyme systems of the cell to be able to distinguish RNA from DNA. A typical nucleotide then is going to have one of these two pentose sugars, either the ribose or the deoxyribose, depending on whether it's DNA or RNA. Coming off of carbon number 5, you're going to have a phosphate group. And coming off of carbon number 1, you're going to attach one of these nitrogenous bases, the pyrimidines and the purines that we mentioned. In DNA, there's the 4 different options for the nitrogenous base. Could be cytosine, thymine, adenine, or guanine. In RNA, the four options could be cytosine, uracil, adenine, or guanine. The thymine is not available in RNA, nor is uracil available in DNA. And what ends up happening is these polymerize together, again through dehydration synthesis, from the phosphate of one, and here's that beginning phosphate. We call this phosphate because it's coming off carbon number 5. We call this the 5' end of the strand. We've got a phosphate going to a pentose sugar, and then coming off of carbon number 3 right here, which if you look at DNA, for example, that's this hydroxyl group right here. We're going to go through a dehydration synthesis between this deoxyribose and the phosphate group of the next nucleotide. You notice the nitrogenous bases aren't actually involved in linking together. So we're going to go from phosphate to sugar from one nucleotide down to the phosphate and sugar of the next nucleotide, to the phosphate and the sugar of the next nucleotide, and so on. And so we call this the sugar-phosphate backbone. If the nitrogenous bases were here, there would be no information, because all we'd have is sugar, phosphate, sugar, phosphate, sugar, phosphate. It'd be absolutely identical all the way down the line. It'd be like if I gave you an A and a B, and I said write a really long word with those two, and all you can do is alternate them. Well, the only thing you come up with is A, B, A, B, A, B, A, B, A, B, A, B. There's no variability. There's no way to create novelty. So by attaching these 4 different nitrogenous bases, we can get all kinds of different patterns here that spell out words, in a sense, that tell the ribosomes and the rest of the translational machinery how to create a protein. Now this polynucleotide strand, if this was just a single strand like this, in most cases that would be an RNA, because RNA generally exists in a single-stranded state. If, however, it's a DNA strand, then we're going to get 2 of these polynucleotide strands in reverse orientation to one another that are following what we call base pair rules to bond to one another with hydrogen bonds. If you need to review hydrogen bonds, go back to episode 1. But here you can see there's our 5' phosphate end, and way down here at the end of this strand is our what we call our 3' hydroxyl end. So this orientation is from 5 to 3 as we go down. The complementary strand, however, here's its 3' hydroxyl, here's its 5' phosphate. Each nucleotide is flipped around and the whole strand is upside down relative to the first strand. So if the strand on the left is in the 5 to 3' orientation, the strand on the right is in the 3 to 5' orientation. And then the nitrogenous bases are in the appropriate orientation when these are flipped around like this to reach one another and to be attracted by hydrogen bonds. T's and A's, thymines and adenines, are attracted by 2 hydrogen bonds. Guanines and cytosines are attracted by 3 hydrogen bonds, and so are a little bit stronger. Each of these hydrogen bonds is pretty weak, but when you string thousands and hundreds of thousands and millions of them together, it becomes a pretty strong network that holds the 2 strands together. And just the physical nature of these strands, they've got a little bit of a kink in them. As the nucleotides reach one another, it twists them into this famous double helix that we're all familiar with. Now, if you think about these nucleic acids as information polymers, consider a gene, for example, that has 1,500 base pairs. And that's pretty typical for a single gene, a single recipe for making a protein. 1,500 pairs of A's and T's and G's and C's. Because there's 4 possibilities in every position, that's 4 to the 1,500th power possible combinations. And that ends up being something like 10 to the 20-something power, something like that. I'd have to recalculate. I forget it. It's a huge, huge, huge number. One with dozens of zeros behind it, which is more possible gene sequences that you could come up with in those 1,500 base pairs with only 4 letters in the alphabet than there are stars in the universe. That just blows my mind. I think that is so cool that the language of life, DNA, is this language that is, for all intents and purposes, infinite. Never going to run out of sequences to create new and novel proteins. It's really a remarkable thing. All right, let's summarize this review of our nucleic acids. Nucleic acids are the polymers that we think of as RNA and DNA. They're involved in information. Nucleotides are the monomers of these nucleic acids, and the DNA nucleotides are slightly different than the RNA nucleotides, deoxyribose versus ribose. That's the main difference, as well as uracil versus thymine in the nitrogenous bases between the two. DNA forms a double-stranded double helix where the two strands are in reverse complementary orientation. Reverse meaning the 5' 3' ends are in opposite orientation. Complementary meaning that it's following base pairing rules. A's and T's are always paired off with one another through hydrogen bonds, and G's and C's are always paired off with one another through hydrogen bonds. And then finally, your typical gene that acts as a recipe for making a protein is 1,500, even 2,000 base pairs commonly, and the diversity of sequences that we can come up with 4 letters and 1,500 or 2,000 positions in the recipe, in the word, is virtually infinite. Really a remarkable molecule for containing and relaying the information of living things.